Okay, so if the panelists come and join me here and we bring in the fifth chair.
Who doesn't get a chair?
Thanks. Okay, so you've sat listening to us for a while. So rather than me ask the panel some questions, I think I'll turn it over to the audience to ask, to see if there's any burning questions. Anyone?
Philip.
So this is really for the first speaker, Neil. So you raised this sort of question of, obviously, of lack of confidentiality in your compound being given to the next door lab that then published. This actually raises another question and problem which I'm trying to solve at the moment, and which should have been solved long ago and actually would help in such situations, and that is, I presume, of course, you don't supply the structure of the compound to the academic collaborator, and therefore the colleague it was given to didn't know the structure either, but he was able to publish. Now, I've had a long-standing battle with many journals and editors to try and persuade them never to publish a paper that uses a compound without a structure. Because if they do such a thing, it defeats the whole object of publication, which is to put it in the public domain and to allow people to test its validity. Mm-hmm. Obviously, if you don't know the structure of the compound, you can't therefore use the compound. That's impossible. So this problem actually could be at least partially solved. I've just realized that this is another reason why journals should not publish papers with compounds without compound structures. Yeah. Because by doing so, they would actually, in addition to everything I've told them so far, also, protect the company's compounds, which may have been misused. Maybe you'd all like to comment on this sort of
yes,
issue.
No, it's a very good question, Sir Phil. My first thing to say is it's not Simon, by the way, who I'm talking about. You are right. It is something we sort of struggle with a wee bit, because when we're quite early into our drug discovery process, and we want to get our compounds out there into the academic environment as fast as possible, because we feel that publications would be helpful, new science generation would be helpful, but we haven't quite nailed the IP on the compounds, and it becomes a sort of balancing act between trying to get the compounds out there and trying to protect them. So we often think about, have we got a probe compound? Have we got a compound that we can send early? Because we understand that nowadays, really, if their academic is going to publish, they like to publish the structure with it. And can we actually give that bit of information up when it is required for the publication? So it is something we do struggle with, Sir Phil, and I think that when you're early on in a small company and you're trying to get as much science around that project as possible, having collaborations is great, but they do have their downside in some respects.
Personally, I never worry about whether I can't publish with a company compound, because if I know the answer, I can get the answer using another technology subsequently, and then I can publish the same result without using the company compound, or at least by and large you can.
Yeah, not with ours you can't, Phil, because no one else has dub inhibitors as we have.
No, I'm saying that there are other technologies besides using inhibitors to achieve the same research result. Yeah. So I've got on a sort of slightly similar but not quite such the same vein with something that Simon mentioned, that he used the combination of the RAF inhibitor and the MEK inhibitor to overcome drug resistance. And the paper was published in Life Sciences Journal in 2011. That actually is a good argument for companies not allowing academics to publish, because, as you know, this allowed GSK to get in first by combining dabrafenib, the RAF inhibitor, and trametinib, their MEK inhibitor. And as a result, which of course was approved for combination use last year. So, so actually, AstraZeneca actually damaged their situation by actually allowing that paper to be published. So it's just a sort of a comment, you know, and that this is always, there's still extreme danger for companies in publishing, even something that looks relatively innocuous at the time.
I think AstraZeneca knew that train had left the station by then anyway, and so I don't think they had any problems with that at all. There's another speaker, question up the top there.
Yeah, my name's Rob Pinnock. I work for Merck Sharp and Dohme. So this is an informatics question, so I think it's for Jeffrey, but the rest of the panel, please chip in. So we heard this morning about data quality in the literature that's in the public domain, and you're mining public databases. And I guess there's an assumption at the moment that OMIM and MCBI is actually correct. Is that true? What can you put in place to actually mitigate the fact that some of that data might actually be wrong?
So I think it's a great question, and there are two angles we're taking on it. So one is to try to separate the things where we can really, with some statistical confidence, say we believe in these things. So a lot of the genetic data, the modern genetic data, I mean the sort of candidate gene literature was full of crappy stuff that, you know, was really confusing for a long time. But I think the academic genetic field has got to a place where we really can say these are the things we're incredibly confident of and we know are right. And one of the things that CTTV wants to do is to pull all of those in and put them in front of researchers in a way that separates them from necessarily then all of the rest of the literature. Now, you don't want to throw away the rest of the literature, of course, because there's a lot of useful information there, but even that starting point of one of the ways I think about it is you want to get to a point where as much as possible there are automated tools that someone can use that are almost as good as sitting next to an expert who can kind of, you know, talk you through which bits of the, say, you know, again, focusing on the genetics, which are the most important to pay attention to. Now, the trouble is, of course, there's then a huge raft of other types of data, so maybe you care about expression data or something like that, and that has a whole larger history, which is probably a bit messier and hard to say which is good and which is bad. So on that side, I think we're trying to start with the sets of data that we can be the most confident in, while piece by piece bringing in additional types of data that, you know, scientists really want to be able to look at, but we don't yet have as strong an idea of how to completely curate them, the sort of best from the not so good. But it is very much a live thing, and so it's, you know, it's something we're trying to work our way through. If anyone else wants to?
I think the only thing I'd add is that bioinformatics, I think exactly the same rules apply as to wet biology. If you put crap in, you'll get crap out. Yeah. And I think the best bioinformatics and the best systems biology has been done by bioinformaticians who work closely with wet biologists who actually have the knowledge to say, don't trust that paper is rubbish.
Yeah, and another thing, just sort of touching on that, is it's a hard problem. It's an incredibly stupid solution for every company to try to figure out how to deal with that and sift the wheat from the chaff themselves. That's one of our founding things, is let's try to bring the people who can talk to the experts and at least try to do a good job once rather than a mediocre job 10 times. There's a question. Yep. Yes.
Thank you very much. John Wise from the Pistorius Alliance. The title here, De-risking Partnership, Sharing Data in Collaboration. Clearly, as the industry gets more into this externalized R&D mode, sharing the data becomes increasingly important. So I guess my question is in two parts. One is, do you think senior management looking for this open innovation, externalized R&D paradigm, hoping that is going to bring the productivity in R&D, pays sufficient attention to data sharing, the technologies, the disciplines, the standards of data sharing? And secondly, do you think we have data standards for effective data sharing in this externalized world of contemporary R&D?
So to the first question, I think it varies hugely. So, you know, GSK at a fairly senior level, I think, believe in that idea and got behind it. And, you know, I've spoken to other companies, and some really fundamentally, I think, aren't interested. You know, they don't see it as a commercially sensible step to do this open data sharing and to engage in things where they would have to share so openly. Just as an aside, I mean, GSK obviously doesn't share everything with us. All of the work they do with their compounds is completely protected, and they sort of, you know, they see the benefit is where they can bring their internal, mostly chemistry-focused work up against the sort of the biological target data we're putting together. So it varies on the one hand, I would say. And then this question about standards, I mean, the short answer is no, we definitely don't have them yet. It's a very important problem. There's a thing, again, just from the genetic side called the Global Alliance for Data Sharing and Health. It has a very grand name. It was put together to try to address some of these problems of making it possible to share data in a way that's productive without necessarily, because there's always restrictions about, well, we can share in some sense of the word, but can't physically put the data all in the same computers in this country or, you know, under these restrictions. And so I think there are efforts to try to make as many different types of data interoperable as possible. And I think that's the important thing, is it might be impossible to ever get it all in one place. What's important is that if I have some valuable resources here, that you have a way that you can access them and you know, compare them to data you have there in a sort of fruitful way, and that it's not some kind of a, you know, a standards thing that just stops that from ever happening. And it's definitely not a solved problem now, I'd say.
Yeah, I'd say as well, I mean, I think senior management think about these things quite a lot. So I think that they probably have, but we also, not all data sharing is open. And not all open things involve sharing all of the data. I mean, you essentially just said that. I mean, the open innovation, I know it's called open innovation drug discovery. The open part of it is opening the set of assays to anyone in the world who wants to, you know, fill in the form. But, and although they share their compounds, they don't share the structure of the compounds. So this is where, you know, what you share. And we come back to the IP and because patentability, obviously that's extremely important. Anything that is in the public domain is no longer patentable if you haven't patented it already, obviously. So an awful lot of when we complain about our lawyers, we don't really mean it, because the lawyers are the ones who are making sure that things remain patentable, but for both sides. Both, you know, at a small company that we might be talking to, or or academics, and for us, for our own, ensuring that we don't get contaminated, and the good work of the scientists in the Lilly labs is also protected, so that they can patent the things that they've made. So there's the open and sharing, and you know, and then I've got another example for you of sharing standards and data standards, which is a little bit perhaps further away from these early drug discovery, sort of early development, but in the later stage drug development, which is in fact where the huge investment comes. It's where real money is being spent. There is an initiative which was probably on my wheel of different collaborations, which we and I think most of the big pharma belong to, where essentially we are sharing data with each other and protocols and practices around qualifying clinical sites and all the different processes that go into setting up and running a clinical trial. Because if you are, you know, I don't know, an Alzheimer team, neurologists in a hospital in London, and you want to run a trial with AstraZeneca and another trial with Lilly, you have to go through a whole series of the same, basically answering the same questions about how you manage your data and patients for everybody. And so the idea is to put all that together, so you only have to do it once. So like, you know, you get your, it's probably not, you know, you get your MOT, as it were. It's not called that anymore, is it? For your centre, and it's valid for every pharma company that signed up to it. And a series of similar initiatives around how you store data and how you, so that it's all transferable, with the idea that one day it'll all end up in something like you're doing. So it's happening.
And I think on the cancer front, we're seeing a move to want to have a sort of single set of validated tests that different pharma companies can access. So they're all accessing the same test, but then the information that is made available to them is on the particular genes of their interest. So you can basically make one large panel but actually you don't read out all of the information from that panel because it doesn't have all of the genes that, or it contains it contains the superset of genes that those companies would be interested in, and you make available the subset of those genes that those companies are particularly interested in. That's something which seems to be gaining a lot of traction at the moment as a way of trying to form one common standard for how to generate genomic information for cancer biology.
I think this is also very important in the academic sector, and we're seeing this more and more. And a very good example of this actually is the work that Philip and his colleagues in Dundee have done on kinase inhibitor profiling. So they've published several papers updating on the field on the specificity and selectivity of protein kinase inhibitors. And that is now an enormous resource that myself and many other people turn to again and again. And there's actually a great website where you can go and type in your particular inhibitor of choice, and it will tell you actually all the other kinases that it inhibits at this concentration of ATP and at this concentration of drug. And those kind of data standards are becoming increasingly important, and that is a very good example of leading the way.
Other questions? Okay, I have one then. Oh, there is one?
Just one
okay,
last.
yep.
So I just want to know, like, what is the difference between the academic research and the drug development pipeline that is going on in the industry? Because, you know, when we were discussing in the morning that let's go from personalized to individual therapy. So, you know, when we look at the cancer, you know, even in individual, the cancer is really heterogeneous. A few population might express, let's say, EGFR, others might not. So the data that we generate using cell lines in the academic research, and I think that's the fundamental lead that these companies perhaps take and develop these therapeutic molecules. You know, so so there are recent reports that these cell lines are, first of all, they are really homotypic. They don't have any sort of heterogeneity, and moreover, there are many cell lines which these academic groups are using, which are by genomic sequencing, they are shown to have low-grade cell types, but they are used as a high-grade or metastatic cell types. So, what is the authenticity of these sort of academic research? And the question is, like, because these pharma, they are making these lead molecules and they are really working in the clinic. So, what is the approach that these pharmas are taking differently than these academic establishment, if at all?
So I think this comes down to the quality of data that's been produced within both environments, I suppose, is your question, and whether when you're looking across cell lines in oncology, you know, what that data is actually generating and how good that data is. And I think that's certainly been a learning for the industry as we have gone forward in terms of the type of compounds and the quality of those compounds that are being delivered. Because, yes, when you look at stuff in a 2D plastic environment, which is a cell line from a tumour, it is not the same as a human cancer at the other end with the heterogeneity that you're talking about. But I think it touches on both academic and industry. I don't think it's a particular problem to either environment. But being aware of it is important, and having mechanisms that you look at the quality and you understand the quality of your product as you develop it from those early experiments on cell lines is very important. And modern techniques such as the patient-derived xenografts and all those sorts of things are very helpful in adding to that quality. I don't know if that was the gist of your question, but I think that's probably what I got from the question.
Hi, thanks for a great talk. I'm Gary Parado from AstraZeneca. Jenny, I wanted to ask you, you showed that lovely map of all your open innovation collaborations, and clearly Lilly's kind of led the way with this and has done it for a really long time. But I was really struck that they pretty much all seem to be in North America and Europe. Is that a concern for you? Was it a strategy? Are you missing an opportunity?
I was living in Canada until very recently, so I got quite upset about the fact that there were no bubbles in Canada. And then I realized that in fact there are. It's just you can't, because they're all those little bubbles from the northern United States actually go over the top of most of the Canadian ones. So there are, it is, but you're right, North America and Europe predominate. There's a couple in Japan, but they're all in the same place. I mean, there's more than a couple, there's like 10 in Japan, but again, so a certain amount of it is data display. But what you would expect is a few more from Latin America. And we have tried, or at least it's, again, there's a group of 4 very dedicated people who put almost all of this together. They have tried really hard. And it's extremely difficult to get sensible, well, not necessarily sensible, but MTA agreements. Because the way that it works is that the institution signs an MTA, it's an academic, when it's an academic institution signs an MTA with Lilly. And this seems to be a very difficult thing to do in most Latin American universities, to come up with a language which is broad enough to cover every employee, whatever, academic from the institution, and yet will fit with what, you know, our Lilly legal department feels is necessary from the liability IP point of view. So it's just been such a struggle. And I don't know why we have so much trouble with that, but I guess it's perhaps a different legal system which isn't as flexible. The rest of the world, I think it's not so surprising that we're not seeing so much from. And perhaps it's also we don't have as much reach in places like South Africa and so on, where I'm sure there are people doing good chemistry.
That's really interesting that it's a legal thing, not a science thing.
No, it's a legal, perhaps cultural as well, you know, understanding the... I mean, again, it all comes down to people and how the different sides view what you're trying to do with this.
Yeah.
So.
Okay, thanks.
But they're working on it.
So, a question for our panel, and maybe for people to think about in the audience as well. So, we talk about people as being key to driving these collaborations and the need to build trust. And if you're a small company, you want to move really, really fast. How do you feel comfortable in that, going from 0 to trust and basically handing over your compounds? Do you just rely on the fact that you've got the legals to protect you, or what steps do you take to establish that whoever you're talking to is going to be somebody that you could actually build a trustworthy relationship with?
Yeah, it's a good question. It often comes down to the fact that we know the scientists on the ground, so we have established already a relationship. And I think, as Simon says, relationships are very important. So there's a mutual respect around the science. And then I think that then generates the next phase, which is, you know, getting your compounds, getting the sort of science and the research agreement sorted, and getting your compounds out there for them to test. So it's got to have a basis. And I think if that basis isn't there, sometimes it is quite difficult to then see where your agreement or where that plan is going to go. That for me is important.
So it starts with the scientific common interest.
It does start with the scientific common interest, and if that's missing, then you almost feel as if they're wanting just to take your asset and close you down tomorrow sort of idea, rather than have a research with you.
Jenny, how would you respond to that? At the other side, you know, the side of the coin, being a much larger company.
Yeah, but in the end, I think it's establishing relationships with small companies. Also, there is an element of track record. I think that I find, I mean, this is people's comments to us, is that if you are, I think different companies work in different ways. And if you've been working in a field for a long time, you've probably got quite a lot of these experts and scientists in the field who are out there in the environment making connections with the small companies, and naturally from a scientific point of view, not because they're business development people. They know, you know, you may have sort of world experts within the company, and that helps because then you have a track record. When you're moving into a new field, it is, it's, you know, getting out, in a way we as the larger company, the partner, have to sell our skills, you know, demonstrate that we can offer something to a, and the ideal partnerships are those with a small company where, you know, both sides are bringing something important to the overall result. And I guess there are other ways of doing it, which is there are some companies, some small companies, that truly want to, you know, hand you their molecule because they want to take the money and do something else. I mean,
Money
not
and run.
Money and run, not necessarily run and, you know, buy a house in Marbella, but run and, I mean, but I've done one deal where they said, you're the experts, you take it, we are going to found a new company now, because we like doing discovery.
Yeah. You're right, it does come down to the philosophy of the small biotech and what they're after out of the deal. Mm-hmm. You know, are they a long-term company, or are they, you know, one product asset, get rid of it and move on type company? So that can structure it a wee bit, yeah.
And I was going to ask Jeff the same question, because you've obviously formed the public-private partnership with GSK, and the intent is that that will then expand to take on other partners. So having been partly kind of molded in GSK's form, how are you, or to GSK's purposes or need, you now want to go, or will want to go and form partnerships with other companies. How are you going to establish that trust there with them?
So, you know, the honest answer is it's a problem we haven't fully solved yet. Sure. I mean, as I said, GSK, they considered, I think, trying to form a consortium to start this, but feared that 2 years later we'd still be trying to work out the politics of negotiating with 5 companies and so forth, and they wanted to get going. And I think that's probably a sensible thing. You know, I've seen some examples of, for instance, the Accelerating Medicines Partnership in the US is doing a lot of cool stuff, but has a lot of sort of mass in terms of getting all of that stuff on board and started. So I think we're trying to find a couple of different ways. I mean, one thing is not everything we do necessarily has to involve all of the partners. We do sometimes get talking to, you know, company X that, oh, but then do we have to talk to GSK? And it's like, well, not necessarily. On the other hand, I don't think a good outcome for CDTV is just that Sanger and EBI have separate, you know, relationships with 5 companies that never intermingle, because that's just, I think that's missing an opportunity for something bigger. You know, one thing about that trust that has become obvious early is different companies have different core therapeutic areas, and if there's a little bit of, if there's less overlap, then I think people just feel a bit more relaxed. You know, GSK isn't going to develop a drug in blah because it's not one of their core areas. And so that's one thing that, you know, might help us get moving in the early days. And then the other thing I would say is just, you know, we do want to, in many of the things we're trying to do, kind of create generic resources that will be really interesting for one reason to one company and for a totally different reason to another company. And that, again, makes people just feel a bit more relaxed in terms of building up that relationship of trust. And I mean, I think that word is the one that we keep hearing and is obviously essential. And I have to say, I don't have a magic solution for how you build that. It's not an easy thing.
Simon, anything?
I guess from my perspective, I think part of the reason that our collaboration with AZ has gone so well, and hopefully with Mission and Aztecs as well, was I spent 6 years in biotech in California before coming to Babe and doing drug discovery. And although I've chosen to work in the light side of the force now rather than the dark side of the force, I have that perspective, and I have an appreciation of what drug discovery involves, and I also have an appreciation of what are realistic expectations that academics could make on their pharmaceutical or biotech collaborators. And as well as my day job running my research group, I'm also kind of, as you alluded to, responsible for knowledge exchange and commercialization within the Babraham Institute. And we're kind of trying to kind of institute a process of, as it were, educating my colleagues to have the same sort of mindset, that actually working with pharmaceutical or biotech sector can actually strengthen their research, because it can give them access to unique reagents and things like that. And so they should therefore think about the expectations and think about what they want to get out of it, sort of thing. So.
I was really struck by Sir Philip's comment earlier this morning, where he was saying how his institute delivers on their promises on time. And I think there's nothing like that for developing trust, and nothing like not doing it for testing the trust in a relationship as well. We've got time for one more question. Anyone? No. Okay. It's tea time. During tea, we've got half an hour. During tea, there are some exhibits outside as well, so I encourage you to go and look at those, as well as the artwork. So we have an artist in residence, and encourage you as well to go and look at the artwork on display. Thank you very much.